the ImmuZoom seminar this week. As we mentioned before, ImmuZoom seminar is a series of seminar that are organized by many research investigators among us across different countries and regions. And our goal is to really facilitate open discussion beyond geographic boundary and for everyone to get connected, also get reconnected with the new and old friends. And hopefully one day some of the exchange ideas can become fruitful collaborations through this nice platform. Today, I am really excited to host our speaker, Weiqiang Chen from NYU Department of Biomedical Engineering. And I think as we all appreciate, immunology is becoming one of the testing field for multidisciplinary research. Weiqiang will tell us more about how the tools and systems he has developed in his lab and also across his field has revolutionized immunology research and cancer research. Weiqiang studied physics at college in Nanjing University back in 2005 and went on to obtain electrical engineering from Shanghai Jiao Tong University in 2008 and then from Purdue University in 2009. He then earned his PhD degree in mechanical engineering from University of Michigan in 2014. Then he has studied his own lab at NYU and is becoming associate professor at the Department of Biomedical Engineering and the Department of Mechanical and Aerospace Engineering. He has a really impressive list of achievements, among them including Biomedical Engineering Society Cellular Molecular Bioengineering Rising Star Award and Young Innovator Award, the New York Academy of Science Early Career Investigator Award, the NIH Outstanding Investigator Award, the Lab-on-a-Chip Emerging Investigator Award, and many others. So let's welcome Weiqiang. Look forward to your talk.
Okay, thank you, Xu, and thank you organizing this great seminar. This is not my first time attending seminar, but as a speaker, I would like to introduce our research on these microengineering biosystems, the tools for the cancer immunotherapy study. So as I'm sorry, I myself is not an immunologist or even not a really expert in biology, but we really want to develop this microengineering, more like microfluidic systems to study cancer microenvironments and monitor immune responses from the patients, detect cytokines like that. And also we are very interested in the cell mechanobiology, and we want to study the cell mechanics, ECM stiffness or nanostructures, and the force between the T cells or the tumor cells, and how these mechanical properties and the dynamics are involved in the cancer therapy. So first of all, I just want to briefly mention the ultimate goal of our study is to transform the current cancer research platform, like the image showing on the left, which is very complicated with a lot of equipment and devices, into smaller and more functional and advanced systems called lab-on-chip systems, organ-on-chip systems. So I would describe the two applications in the following slides. And we want to develop systems for the cancer diagnosis, modeling, and hopefully testing new drugs for treatment. So the organ-on-chip is new techniques have been developed in the past decade. It's mostly used to do three things. First of all, the goal is to hopefully to replace the animal models as an alternative solution to test drugs or to model the disease or like an immunotherapy screenings. And the second goal is to speed up the testing process. For example, we hopefully can shorten the testing for different drugs using high throughput systems or with multiplex measurement functions to speed up drug testing. And then lastly, to achieve a personalized therapy using this kind of organ-on-chip technology. And then for today's talk, we are mostly interested in the cancer on a chip for the cancer modeling. And the main reason we think it's very useful for the cancer immunotherapy study is because it can recapitulate the human tumor microenvironments and also to recapitulate the human immune environments and the responses to the immunotherapy. It also provides the functionality for real-time imaging and monitoring of the immune cells and molecular interactions like the cytokines. And also, we can still do the traditional assays using off-chip testing. For example, we can collect cells for single-cell sequencing, flow cytometry, do staining on the chip. So it's a more advanced and alternative to the animal models to test the immunotherapy. So we developed two types of cancer nanochip in our lab. So the first example is the glioblastoma for the brain cancer microenvironmental model. The background here is the glioblastoma is one of the most common and aggressive brain cancer. So the current study on the tumor microenvironment have revealed this important role of the immune cells, like a tumor-associated macrophage, in the treatment to regulate the treatment. And only targeting tumor cells itself, the chemotherapy and the immunotherapy may not good enough to achieve the best effect. So our idea is to recapitulate the vascularized tumor microenvironments together with the immune cells like tumor-associated macrophage and glioblastoma cells on a chip. So in this system, we want to study how the immune suppression resulted from the tumor-associated macrophage and how this immune suppression will suppress the patient response to the anti-PD-1 checkpoint-based immunotherapy. I may skip all the details about the biological studies, but we basically achieve the function of mimicking the glioblastoma microenvironment with the artificial perfusable vessel, and we can reconstruct the architecture of tumor cells and ECM, the brain mimic hydrogel. We use the hydrogel, brain mimic hydrogel to recapitulate the ECM and also the tumor-associated macrophage. And then we can monitor the immune cell, especially the cytotoxic T cells and the macrophage in the environments. And we tested different subtype of glioblastoma patient-derived cells on the chip, and we found out indeed this significant difference in the immune suppression and also the T cell activation and the killing efficiency in our system. So this is just a representative image and tell us images showing how we can monitor a single T cells in the tumor microenvironments, and we can monitor, for example, we can quantify their migration speed or distance or the trajectories, and we overall tested different nine different patients on the chip, and we tested these three subtypes of proneuron, classical, and the mesenchymal subtypes of the glioblastoma patient samples on the chip. And we found out the tumor-associated macrophage tended to be more M2-like phenotype and cause severe immunosuppression. And the different subtypes, like the mesenchymal subtype, may have a more severe immunosuppression in the environments, so that they have a weaker response to the anti-PD-1 immunotherapy. And with this idea, we tested the anti-PD-1 and anti-CSF1R co-inhibition on different patients, and test all these testing conditions in our system. And we found out indeed the dual inhibition, I forgot to mention the anti-CSF1R is to target the macrophage, and we can suppress M2 polarization and hopefully can reverse immune suppression in the environment. And indeed, we found out the anti-CSF1R treatment can suppress M2 like CD163 macrophage, and also to increase the T cell infiltration in the tumor and activation in the environments. And we tested the 9 patients in our system, and we found out most of the patients responding to the dual inhibition and shows improved patient responses to anti-PD-1 immunotherapy. But they still have a lot of issues. For example, it's not all the patients are responding as well as we expected, especially the mesenchymal patients. They showed a more significant improvement, but because mesenchymal patients have less response anyway, so after the dual inhibition, their response still not good enough. So here we just want to prove that we can use our biomimic cancer-on-chip system to test the cancer immunotherapy. In particular, here is anti-PD-1 immunotherapy for different subtype of patients, and we can do a drug screening within our system. And hopefully we can identify more biomarkers or targets in our future research. And then we are right now we are interested into clinical implant our system with clinical trials to test the different drugs in the ongoing clinical trials at NYU. So the second model we developed was for the liquid tumor, the leukemia. So we developed a so-called leukemia on a chip to mimic the bone marrow microenvironments and to test the chemo and the immunotherapies. So a quick background here, the acute lymphoblastic leukemia is one of the most important liquid tumor and the most common cancer in children. And mostly they are initiated in the bone marrow due to overproduced immature or dysfunctional lymphocytes. And 85% are initiated with the B cells. With this idea, we want to reconstruct the bone marrow microenvironments with biomimic architectures and cellular compositions in our system. So you can see from the schematic here, we reconstructed the central sinus and the perivascular niche with endothelial cells, mesenchymal stromal cells, and the leukemia cells. And we also generated the outer layer, the endothelial niche with osteoblasts. And we can see from this image in the middle and on the right, the in vitro device structures have very good similarity with the in vivo structures. And in the first few slides, we are just discussing the in vitro system for the stroma environments and how the stroma environments affect chemotherapies. Again, similar to the solid tumor model, so we can also monitor how the cell can migrate or infiltrate in the system, and we want to study not only the treatment but also the progression of leukemia formation. And we tested the 3 major chemotherapy drugs for leukemia on our chip, and we found out indeed with or without the stroma microenvironments, they have significant difference in the response to the chemotherapies. So we proved here overall the chips with the stroma environments have a resistance to the chemotherapies. And we want to study in the details how the different cell populations and through which signals they may protect the cells, the leukemia cells from chemotherapy. So the overall hypothesis here, we screened the different signalings. We identified the cytokine signaling like CXCR4 and CXCL12, and the adhesive signaling of VLA4 and the VK1 are the 2 major signalings we want to test on our chip. We do find out the endothelial cells can maintain very stable CXCL12 expression, while mesenchymal stem cells have a decreased CXCL12 expression. And also endothelial cells have increased VK1 expression, but on the other hand, mesenchymal stromal cells increased OPN expression. So in short, we found out overall mesenchymal stromal cells shows a more important role in regulate the leukemia dormancy and may cause the resistance to the chemotherapy. And not only the major stroma environments overall protect the cells, leukemia cells from chemotherapy, but in different regions or locations, they have different effects. So we found out a special difference in terms of in the endothelial and endosteal niche, again, the cells near the endosteal niche have a lower killing rate by the chemotherapy, which means this endosteal niche demonstrate a stronger protection effect to the leukemia, to the chemotherapy. So we concluded here, we tested another combination therapy with different drugs, anti-CXCR4 or VIL4 or NF-kappa B, which is downstream signaling for the two other signalings. So we found out indeed the stroma environments can improve the chemotherapy response, like we show here in the in vitro on-chip study, especially the anti-kappa B treatment combined with the chemotherapies more significantly improve the patient response. And we also test a similar combination therapy in the animal model. We observe similar effect for the combination therapy. So this is just an example. We built up another type of cancer on a chip, especially for liquid tumor in the bone marrow environment. And we focus on stroma environments first for the chemotherapy. And we are also interested in the immune environments and immune populations in the bone marrow. And I want to study how these immune populations and signals may affect the chemotherapy as well as CAR T-cell immune therapy. Our collaborators, they recently just screened the bone marrow microenvironment of leukemia patients, and they found out the monocyte and macrophage population may be critical cell type during the leukemia chemotherapy. So with this idea, we tested it both in vitro and in vivo to contacting the macrophage on the chip, and or in the animal model, we proved that indeed the contacting macrophage in the chemotherapy can improve the treatment response. So with this tumor microenvironment model, we want to study immunotherapies and for the CAR T therapies, especially for the relapsed leukemia patients. And we already know that for the relapsed leukemia patients, CAR T cell immunotherapy is emerging and a very effective treatment strategy. But unfortunately, still about 50% of patients may not respond to the therapy. And it's not very predictable which patients may or may not respond to the treatment. So our idea, just like showing the video here, is actually it's a video taken within the device, so we can monitor, real-time monitor how the CAR T cell infiltrate and kill the tumor cells, and model the CAR T cell therapy process. And again, we have the perfusible system. We can load the CAR T cells or even other immune cells into the blood vessel and let this CAR T cell circulate and load into the tumor environments. So here just two videos showing that we can monitor how the CAR T cell escaped from the blood vessel and how actually the red cells are the CAR T cells and the green cells are the leukemia cells, how the killing process happen in real time. And we can also quantify the killing results, like the leukemia cell viability for CD positive or negative leukemia cells. And indeed, we only kill the CD19 positive leukemia cells, but not the negative cells. And we can also monitor the killing process over a period of time, like a week or even 2 weeks. Like we show this leukemia viability. Actually, this leukemia burden size in the system. And we can also, again, as I mentioned, we can do the routine like standing off-chip flow cytometry or ELISA testing or cytokine profiles in the leukemia environments. And we indeed improve that. We prove that in the leukemia chip, the CAR T cells are successfully activated and expanded and can kill the cells, leukemia cells. And the CAR T cell killing activation and killing process also initiate enhance the cytokine profiles in our system. And then potentially we're also interested in to study the CRS, how the CRS may happen, or if the CRS may or may not affect the treatment. Okay, so that's just two major examples for the solid and liquid tumor modeling and the immunotherapy studies. And then we want to also develop a microfluidic sensing system, like sensors for cytokine measurement. As we know, we can measure the cytokine concentration either from the blood for bulk analysis, or we can isolate different subpopulations to measure the cytokine secretion from different immune cell population, or we can capture a large array of single cells from the tumor and to do a single cell analysis. The major sensor we are using now in our lab is so-called localized surface plasma resonance-based plasmonic sensor. So the mechanism here is if we excite a nanoparticle, and there will be emission light also escaped from the particle, and it depends on the particle size and the optical properties. They have a fixed frequency or wavelengths for the excitation light. And when the nanoparticle can capture a small molecule like the cytokine molecules on top of the surface, and it will enlarge the particle size and also change the optical properties surrounding the particle. So we can see an increased intensity and a slightly shift in the peak wavelengths. So with this idea, we can pattern the nanoparticles on the glass substrates, and each small pattern can be used as one sensor, and we can functionize all the sensors with different antibodies to capture different cytokines. And because we are using the imaging method, dark field imaging method, and also we don't use a secondary antibody to label the cytokines after capture, so it can be used for real-time and label-free sensing. So we can monitor, for example, the calibration curve shows that we can monitor cytokine concentrations over the time in cell culture medium. So if we integrate sensor in a microfluidic chip, we may able to monitor the change of cytokine concentration during the therapy process. So we just validate our system with a simple model of a tumor associated macrophage. For example, we can prove that tumor cells can induce the macrophage toward M2-like phenotype, and then we can see enhanced MCP1 and TGF-beta from the M2-like macrophage. And we can, as I said, we can establish a chip with a lot of different sensing bars or wells, and we can detect different cytokines. And we prove that if you add drugs to the cells, and we can see the change of cytokines as well from the measurement. So overall, we achieved a label-free and a very fast detection, like only 30 minutes we can complete the measurement. Compared to ELISA, it's much faster. And also we can achieve like multiplex measurement for a few to tens of cytokines in one imaging process. And we can achieve so-called high throughput measurement from a larger array of single cells if you design a microfluidic chips with all the isolation units. And we can also monitor the cytokine secretion profiles over a time period of time for dynamic measurement. In addition to the single particle-based plasmonic sensor, we also developed a second type of plasmonic sensor called a Nanoplasma Ruler. The basic mechanism is similar, but the difference is we have the two nanoparticles, a pair of nanoparticles instead of using one, okay? Because we know that the two golden nanoparticles if they are very close to each other, their optical properties will change, and we will see enhanced optical signals from the measurement. And if we can use a small aptamer, DNA aptamer, to link them to nanoparticles, and also we design the aptamer to capture specific cytokines, for example, IL-6. And whenever we capture the IL-6 molecule, there will be a deformation in the aptamer and pull the nanoparticles together. So we'll see an enhanced plasma coupling. So with this Nanoplasma Ruler biosensor, we can not only have an enhanced sensitivity, but also we can use a real-time measurement platform to visualize the cytokine secretion patterns from single cells. So you can see from the image here, a single T cells are in the middle, and we have a lot of pairs of nanoparticles, which called the plasma ruler, and we can detect the cytokine secretion from this single cell, and we can see the secretion pattern over the time. So here just quantify the results showing that we can measure two cells and how cytokine secretions over the time can diffuse between the two cells. As I said, the aptamer is a critical component to link the two nanoparticles so that have a property distance between the particle, and after the capture, they can pull the particle together. So how to design the aptamer is a critical process, and it is running by our collaborator at Clemson University, Professor Dingfeng's lab. So we integrate the system, the sensing system in a microfield chip, so that we prove that we can detect the T cells, and we establish calibration curves for IL-6 secretions from the single T cells, and with different stimulations. And we also tested if we co-culture the macrophage leukemia cells and CAR T cell together to see if indeed the CAR T cell can activate the macrophage and then to secrete more IL-6 to mimic the CRS process. So yeah, the conclusion here is we achieved this plasma sensor for either single cell analysis or the previous example, we can detect a large array of cytokines from the either cell culture media or blood. And we can also integrate the plasmonic sensor into a microfluidics tumor microenvironment model, as we mentioned previously. So we haven't done that yet, but we proved such a mechanism, sensing mechanism within a biomimic tumor, not tumor, actually the tissue environments in the adipose tissue on a chip system. So we can monitor how these cell differentiation polarization happens and how cytokine profiles change in under different conditions. So this is our future goal, to integrate the sensing system in a tumor microenvironment model. Okay, so last topic is about this cancer mechanobiology and biophysics. As we always know, the tumor microenvironment have different cell populations, and not only the chemical soluble factors, but also mechanical factors are very important in regulating the tumor progression and also the response to treatment. Right? For example, we know they have a strong enhanced solid stress in a 3D tumor tissue. And if you measure the force or stiffness of each tumor cells, you will see a difference in the tumor stem cells or the tumor cells and the normal tissue cells. We developed a few micromechanical systems as a toolbox, so we can measure, for example, the traction force of cells. We measured the force from the cancer stem cell and the normal cancer cells. Here it is breast cancer. So we measured the cancer stem cell, breast cancer, normal breast cancer cells. We found out indeed the breast cancer stem cells have a lower cellular force. And then we can also measure the deformability of the cancer stem cells compared to the cancer cells. And we found out the cancer stem cell is softer than the cancer cancer cells. And the adhesion property of the cancer cell might also be important, especially for metastasis, right? So we know that cells, the primary cells may have to detach from the tumor and then circulate in the bloodstream and settle down in a second site for initial new tumor. So in our study, we try to use using a microfluid chip. We culture cells in a microfluid channel, and we detach the cells using mechanical flow, using a shear stress to detach the cells. And then we found isolated low adhesion cells and high adhesion cells, and then loaded into the xenograft mouse model. And we found out indeed the low adhesion cells is more stem cell-like, because we didn't really identify stem cells, but we feel like it's more stem cell-like. We skipped the biology here, but we found out indeed that they have high expression for the cancer stem cell marker, and they initiate more and larger tumors in the animal model. Not only the adhesion, for example, we know the cell stiffness, right? We already found out the cancer stem cell might be softer than the cancer cell. So again, without using a cancer stem cell marker, we just collected cancer cells and culture them on a soft, stiff substrate. We know that the tumor microenvironment may change the cancer cells' mechanical properties. So we mimic such a process. We collect a soft cell from soft substrates and stiff cells from stiff substrates, and we found out indeed the soft substrate, the cells from soft substrates shows higher LDH, the cancer stem cell marker, and we also load into the tumor, the xenograft tumor model. So we found out the soft cells initiate more tumor and larger tumors. Not only the single cell properties might be important. So we know that the tumor cells are in the tissue microenvironments. We might not be able to really study the 3D properties in our system now, because if you want to do the traction force measurement or other property studies, it's better to do a 2D system. So we still try to mimic the 2D, the multicellular system in a 2D model. We micro-pattern the glioblastoma cancer cells on substrates, and we measure the traction force from the pattern. And we know that it's not surprisingly, the traction force is larger at the edge of the pattern. In the center, it has a smaller force, because mechanically, you need to balance the pattern with different traction force. So surprisingly, not only force change from the center to the boundary of the pattern, we also measure the 5 major cancer stem cell markers for glioblastoma cells, and we found out indeed all these markers showing higher expression at the from center to the edge compared to the center area. And if you match these two trends together, indeed they have similar trend in terms of the force and also the cancer stem cell expression from the center to the boundary. So I didn't have a conclusion for the previous slides here. So why that happens, we are still working on that. But that's a very interesting observation. So lastly, I just want to discuss one more application of a micromechanical system to measure traction force from the CAR T cells. And we want to functionalize the PDMS micropillar arrays with the CD19 antibody, and to mimic tumor cell surface membrane to activate the cancer CD19 CAR T cells. And indeed, we proved that we can measure the traction force from the CAR T cells on CD19 positive substrates or non-CD19 substrates. So we found out the dynamic response of the cells, CAR T cells on CD19 positive substrates is different from normal adhesive substrates. More importantly, we found out within only 10 minutes, the direction of the force changed from the outward direction to a negative direction, which is inward direction. The amplitude of force also changes kind of achieve a peak, but also saturated, and some of the cells can come back to the baseline after 10 or 20 minutes. But the data is still very not clean. So we have to say this way, we test many cells. We are still trying to figure out the real pattern of the force response. So, but at least we can confirm that the acting, the traction force in the CAR T cell are significantly enhanced within a few minutes, and that will be mimic the killing process of CAR T cells. Not only the traction force or contractile force of the T cells increased after activation, but we also found out the CAR T cell become more mobile. They are migrate faster and with larger distance on the activated CD19 substrates. So that's also interesting observation. We are trying to figure out what's the reason. It might be a few factors. It might be just because our substrates may have some special properties, or maybe just some biology happening there. So we are still exploring the mechanism about why the mobility of the CAR T cells are enhanced by the CD19 activation. Okay, I would like to stop here. As I already discussed, we are interested in the microengineering tools for the cancer immunotherapy study, from the tumor microenvironmental model to biosensors for cytokine measurement and the biomechanical system for the cellular mechanobiology study. So I'm happy to take your questions. Yeah.
Thank you, Weiqiang. This is really, really cool talk. So yeah, Xiaolei, you have a question, go ahead.
Hey, Weiqiang, this is a spectacular talk and really eye-opening, so many really exciting new technologies. So I have a few questions. The first one is about this SPR-dependent detection of the secreted cytokines. So do you think these can detect the oligomerization state of the secreted cytokine? Like if it's possible to differentiate a monomer versus a dimer or tetramer?
I would say right or no. But as I said, it really depends on design. It's very similar to, if you can just imagine like ELISA sensor. If
yes.
you want to test that, then you have to design the so-called optimal antibody, right? If you use a regular antibody, then if you want to, so it's not possible now for our current system.
Yeah, because it's a little, because it's SPR, like, because the mechanism the SPR works is like, it depends on like how big the particle is, so that will change the signal. So I just wonder, like, in addition to like detect the amount of the cytokine that is secreted, whether like this method has some additional like advantage of telling the oligomerization form of the cytokine. I think that might be.
Yeah, I think that's possible, but I really didn't do any work on that. My feeling is it is possible, but really rely on your design, because that's a very sensitive to a few nanometer of size change.
Yes.
So if you think that will be in that range, right, a few to 10 nanometer change will cause a significant optical response. So, but you have to design this kind of molecule banding, right? And then
yes. So for the Nanoplasma Ruler, so that's another exciting tool. So basically, if you like that will enable to monitor the kinetics of the secretion. So basically, you can detect a concentration gradient of these cytokines as secreted by the T cells, right?
Yeah.
And do you usually see it's like a pretty symmetric distribution, or it's like asymmetric?
That's also, yeah, actually we was thinking about that, because we have very limited data now. We only test the cells communications based on the intensity and secondly, some diffusion patterns. Our hypothesis is if you have two cells for them on the left and on the right, they may have a asymmetric sequential pattern, but that haven't been done yet. That's a very good question. We want to test such a kind of condition, only one cell on the left and the other cell on the right. We believe so, but I don't have any data to prove that yet.
Okay, that's cool. So are these like, is there a panel of for the like a common cytokines that is available for these like to be detected by these aptamer or aptamer-based ruler?
We have a few, I think we have 4 to 6 cytokines now. It really depends on, it's easy and difficult. Easy is as long as you have enough money. For example, if you pay some company like $20,000 per cytokine, they just design it for you, right? And we have a collaborator, we just do it in our own lab. So they do the design for us. They do run the simulation. I think it's basically biophysicists. They don't run the optimal design. I would say we can achieve more cytokines if necessary. If we have enough money, basically just pay them. They will do this screening. So we have 4. I think 4 is working. 6. I think the other 2 is still running kind of optimizations for the design and the testing, experimental testing now.
I see. So how specific are these probes? At least based on the 4 or 6 cytokines.
I don't remember the. Actually, that's a lot of data from the simulation and also testing. I think it's from what we tested is for the current cytokines, they are quite good, better than antibodies. Okay.
Oh, that's great.
Yeah, but if you want to test the new ones, I said you have to do all the simulation first and then do testing. So it's just optimization process. Like each one just run the process one more time, and it just costs money and time.
Exactly. Yeah. Do you have an estimate of the detection limit? Like what's the lowest concentration it can?
For concentration measurement, for current system is 18 picogram per ml. 18 picogram per ml. Yeah. Okay. Yeah. And we have it's not our technology. The collaborator on this actually our proposal. We have 2 PIs. The other PI, they have a technology to integrate the electric field on a microfluidic chip. You can enhance the sensitivity to down to 0.1 picogram. Wow. That's really amazing. Yeah, but we don't do that because normally we just care about around ELISA, the 10 to 20. Yes, these are abundant like cytokines. Yes. Yeah, I think the major issue with the sensitivity is not only ideally you can achieve very good sensitivity, but the problem is the diffusion process for the molecule to approach to the particle, sensing particle is very long. When the concentration is very, very low in the medium, right? So it takes a long time for them to be detected. So in our measurement, we just normally detect within 10 minutes, 20 minutes, or 30 minutes maximum. So there's 2 ways. Either waiting 1 day, you get achieve a higher sensitivity. Otherwise, as I said, we just use the electric field to pull the cytokine molecule towards the sensor. So that will be another strategy to improve the sensitivity. Yeah. So basically, like if the sensor is immobilized on the surface, then it will be like more concentrated. Yeah, ideally, it's a single molecule measurement. You can imagine because each aptamer can capture 1 molecule, right? So you can achieve very high sensitivity. But if you ask me the concentration measurement from the cell culture medium, that's like 10 to 20. That's the range we can achieve without any actual, mechanisms. Yeah. Okay, that's great. Yeah, cool. Thanks so much.
Chen, before I go to you, I just want to fill in a couple related questions. I also see one related question in the chat as well. Yeah. Just follow up with the aptamer measurements. This is really interesting technology. One clarification is that is the measurement more about the concentration for the cytokines or diffusion range of the cytokines in the current system?
Can you say it again?
Using the picture you showed, you have the cells, you have the beads that light up near the cells. So do you think there's more measurement for the concentration of the cytokines or diffusion range of the cytokines? Because once it's spawned by the aptamers, we're being sequestered away for the cytokines, right?
I'm not sure if I understand the question correctly, but our nanoparticles are fixed on the substrate. So they are
Right.
not mobile. So because we don't want the particle move, because whenever they move, then the optical signal changes, right? Very sensitive. They are very sensitive. So what we do is one particle link to the glass substrates with chemicals, and the other
Right.
particle link the aptamer to another the particle. So we immobilize the sensor. So I'm not sure if I understand your question. So you want to say if the particle move?
Yes, it's somewhat what I was trying to ask is that when you see a signal that is further away from the cells, that's indication that the cytokine you are measuring can diffuse to that particular distance. There's a higher concentration than the 18 picogram per ml that you mentioned, right? That would be the interpretation. Yeah.
Yes.
Instead of a quantification for the concentration, is more of to see how far away the signal can get to.
Yeah. Because we are talking about sensitivity. Normally, we're just talking about when you wait enough time, how many molecule capture that's a concentration or the sensitivity is. Right? What I'm talking about to improve the sensitivity by changing the diffusion of the molecule, especially at the low density, low concentration molecules in the environment. Then it's not enough time.
That's an awesome technology. I think right now for many of the diffusible other cytokines or chemokines, there's really no a really nice way to look at what's the diffusion range of those molecules from their source of cells. This is awesome technology.
Yeah.
A related question from the chat box, if just to squeeze in one more, is that from Kai Wen Luo. The question is, instead of using gold nanoparticles and aptamers, can we rely on soluble Fv antibody conjugated with fluorophores for FRET, for example? Will that make the system more versatile?
Yes, that's again, it really depends on design, right? You don't have to use aptamer. Actually, you can use antibody even, right? It just depends on need. I think that's possible. The reason we use aptamer because it's more specific to the cytokine molecules, right? We can design it by ourselves. And also compared to the aptamer, it is, aptamer compared to antibody, it's much smaller in the size. It's more comparable to the cytokine molecule, right? The primary antibody normally is larger. That's my guess. But as I said, if you want to use a FRET kind of system, actually we was discussing with another collaborator here for another project, we want to use similar design, use a FRET. Yes, that's possible, but I think that's need to redesign everything with the system. Yeah.
Tuan, go ahead.
Thanks for the talk. It's great. I just have these questions related to this leukemia on chip. I want to ask you how actually exactly, I didn't quite get the machinery. How exactly you put the hematopoietic cells on your designed chip?
You mean the T cells?
That's the part I didn't quite get. Is that only T cell on the chip, or is there's a mixture of cell population?
Oh, okay. Yeah, for leukemia on chip, because we have two different chips. So for the leukemia on chip, we have one system, but 3 different versions. Okay, version 1, we don't have any immune cells other than the only one macrophage we tested. And version 2, we have all the patient or healthy donor derived bone marrow cells, BMMC, the bone marrow monocytic, what's it called? BMMC, all the immune cells population from the donor or the patient. So we collect all the immune cells and put it back to the chip. And the version 3, we want to test the candidate cells. For example, we want to test monocyte, non-classical monocyte, or T regular. So then we just put we remove the immune cells, but only put a few targeting cells. So depends on your need. We can either put everything back to the system or only put a stroma cell. For example, we're only interested in stroma. We don't want to put any immune cells, only targeting stroma cells, or with all the immune cells, or with a few immune cells. So we have 3 types of versions.
So in one thing is when you talk about the patient bone marrow cells, you directly to fractionally isolate the cells and then put them on a chip, or you actually differentiate them in vitro and then put them back?
I don't think we if you patient cell, we don't differentiate them. We just collect them, isolate them, and just put it back.
So it's actually
Sometimes we can just buy it. We just buy, if you, for the routine testing, we just buy some healthy donor cells or patient cells from the company. They just have the BMMC, like a PBMC, they already have these
I see.
cells. Yeah.
So, but regarding different cell population, when using the chip substrate or the chip base are all the same. There's no different, in terms of coding or anything like that, different in terms of if you need to put different cell population on the chip.
That's a very good question. Indeed, there's some slight difference in terms of the cell culture. So if you load the in terms of the ECM, it's indeed that's a very tricky part. You have to really optimize the condition, and then you have to make sure the pH, everything is correct, so that you can culture the cells within ECM. And but the cells, when you add more immune cells or different cells, you have to change the medium to make them survive. So I don't know if I answer your question. Is you have to change the cell culture media to make sure all the cells are favorable.
But how long usually the experiment lasts? It's take days or just hours to complete the experiment?
No, for the chip, we have long-term short experiments. For the chip, it's 4 weeks. For the experiment imaging, you can do just like what do you do for your samples,
sure.
Right? So it's like a few hours, or sometimes we do every day to have measurements. So the chip culture 4 weeks.
But if it's lasts for weeks, even to human cells, I mean, if you don't have the right microenvironment, like you said, you code ECM on top of the chip. I think the most of the monocytes and the macrophage cannot last for like weeks. They're probably gonna, die out. How you actually solve that problem?
Mm-hmm. That's indeed we believe our system is because more biomimic for the environments. So that's why the cells can, and also we have to really optimize the culture condition. I said cell culture media is critical. We found out that indeed they are surviving very well after optimization, of course. A potential issue, as you said, they may not die. I don't think we see the cell. If we see cell dying, then that we are not continuing experiments. A potential issue is how the cells can maintain their phenotypes, right, in the chip. So we are right now we are running this kind of single cell sequencing of at least flow cytometry to make sure is the phenotype changes or not. There's a lot of work to validate the system. For culturing the cells, making them alive, we already solved the problem. But for make sure all the immune cell population in biomimic phenotypes, right, or even the composition of the different cell populations within the group of cells, we are running all this validation cases to make sure the protocol is correct. So that's also kind of optimization process. But right now we don't see big issues to cultures, and one possibility is because our system provides a better environment compared to 2D culture system.
So you're talking about after 4 weeks culture, you actually can recover the cells from the chip and then to validate them and then compare with them. I see.
Yeah. Anytime. We normally sometimes collect from 1 week or 2 weeks or 3 weeks, anytime you want to collect the cells.
But the validation mainly depends on flow cytometry or other.
We are running two things, right? And it's majorly staining, flow cytometry, and also single cell sequencing.
I see. Okay, thank you.
Weiqiang, I have a related question to the bone marrow niche part. It's a really cool model. I think some question a lot of people has is about the heterogeneity of the bone marrow niche. In your in vitro systems, have you looked into the heterogeneity of either the mesenchymal populations or different cell type in your system to see if they can recapitulate some aspects of the heterogeneity people observe in vivo?
We haven't done that yet. Honestly, we have the chip, as I said, the optimization takes a long time. We only have the working immune cell chips, as I said, with host culture just a few weeks ago, Now we have the kind of working device for a few months or even last year, but we are optimizing to a better biomimic system until very recently. So we have to really, and we tested CAR T therapy for sure, but we haven't done any other experiments yet. but that will be interesting study we can do.
And so a related question following Chuan's question about the culturing conditions. Another paper is published. Just curious if you need to supplement any type of growth factors for monocytes macrophage in your culturing condition to make sure they're viable for 3 to 4 weeks.
I think so. I forget the details. I don't remember because it's a protocol. Actually, I don't remember the details here. I don't remember what chemical they put into that. I think only the polarization I think we have to do because we are interested in the non-classical monocyte and the macrophage. I don't recall if we have to put all these kind of chemicals. I don't remember that. Yeah.
Cool.
So I think like for the chip, this is really cool system. Weiqiang, do you think like because there's like other like a 3D culture system like these organoid. So I wonder here for the chip, is a physical confinement play some role in like forming the pattern of these like bone marrow or the brain tissues?
That's actually a very common question. We always get that same question from the reviewer of our proposals. So in short, our system, no, we are not trying to say we are better than everything, but overall it's better than organoids by providing immune environments. So I think they're nowadays they're trying to also include the immune niche into the organoids very, very recently. But before that, they are only they don't even have the this vascular system for them. They don't have the vascular network in the organoid. So we have the vasculature. We can monitor the growth of the tumor, right? And also to include immune populations on the immune environments. That's why we believe it's better than the current organoid systems. And also, I think it's more controllable because we can monitor more details in our system. That's also all the ACEs. I think for the organoids, sometimes I think I'm talking patient-derived organoids, right? I'm not talking about iPS-derived organoids. That's a different. Patient-derived organoids, sometimes they have to subcultures and kind of subculture them right every time, so they kind of not have the biomimic environments and the immune response as we can do.
I see. So for the like a vascular system, so these are like maybe I missed in your talk. So these are directly transplanted into the chip, or it's like formed through self-assembly of the like
from cell assembly. So we use EC cells, primary EC cells to reassemble the vascular
cell. Okay, I see. Okay, great.
All right, awesome. Is there more questions? Right, if there's no more question, let's thank Weiqiang again. It's wonderful, wonderful seminar today. Really learned a lot, eye-opening.
Okay, thank you.